like my parents are Chinese. They immigrated from China in like the early 90s or mid 90s. Yeah, like right before I was born.
So you're used
Carolina? Charleston.
pretty good town here.
It's so beautiful.
I should probably start acting like I'm.
Oh, okay.
Well, I've got 5 minutes. I can
But yeah, I think my advisor is a little worried about funding for his postdocs though. So like one of his postdocs is applying for grants because Fernando like ran out of funding for him. So it's a little stressful. And he's not from the US, so I think it's kind of tricky.
Yeah.
Hi.
Good to you.
I'm so sorry.
Yeah.
We have to apologize. We're part of the country that's fucking up the road right now. Really badly.
Yeah.
Okay, there you go. So you're in over Christmas?
I might be going home. And then I don't know. I need to see if like one of my friends who's also at Stanford right now. We're actually like friends from undergrad, but she's doing her PhD here too. Like she might come visit me in Charleston. I might do like a friends, New Year's.
So our daughter is, as we speak, interviewing a residency here at Stanford.
Oh wow! Hopefully she can. Yeah,
I hope
she
that'd
can
be
be
fun.
reunited with
Yeah,
you.
they gave her a break. She had a pretty complicated story, but they gave her a break in her schedule so she could actually attend this particular session, my session. I sent her all those slides.
Oh wow!
I can't remember. She came the last day lecture that year. That wasn't your year. It was the year after you.
Oh okay. Yeah, I'm not sure if I met her.
So people are showing up.
It's a long 9-hour day of interviews. So like, don't worry, I'll do a special session other than today. So
Oh wow! Wait, 9 hours just for Stanford?
yeah, they just line up a whole bunch of people that want to talk.
She's interested in a research position also, so that kind of. Research and politics. She has a lot of people.
Oh, that's very intense. I remember like Stanford also being my most intense interview for the PhD program.
Course?
From Princeton for undergrad, and then I worked for a couple of years, but then yeah, I think I had 7 interviews with Stanford, and it was the most out of any school.
How do you
Better go for that.
I asked.
Yeah, I might take one. I don't know if it's offered. Whenever it's offered. Just like questions or something. Yeah, but it seems cool. So it seems nice that it's.
That's a good sign.
Yeah. What is the course?
I said, my bias, West Coast, that's the West Coast.
Actually, like, is Stanford a great choice?
Introductory.
I don't really know.
Developing.
But I think that in case they're gonna be like 10 or 11.
Hey Mike, how's it going? Can you hear me?
So,
What time zone are you in?
it's certainly.
I'm in New York right now, at NYU, and I'm about to visit Cornell in a bit. So couldn't be there in person, unfortunately. But want to at least help get the session started. And then mostly I'll let you defer and manage the room if that works for you.
I hope it's gonna be. Yeah.
I don't know if I see Olivier there. If possible, someone could just keep an eye on the chat log in case anybody's bringing anything up. But if it's too much, just to manage the people in the room is okay too.
Hi.
And then Mike, we're just about to hit the hour. Hopefully you got some food and drink already. Probably in another 2 minutes, start with a general introduction of yourself, and then kind of start a talk 5 minutes after. I will probably have to drop out to get on a bus here, but then I'll let the discussion go with the group. And the mic, can you just test just saying something out loud right now just to see how your voice is coming through?
So how's my voice coming through? Can you hear me?
It's a little bit low and there's some background noise, but it kind of works. There's an option on the console too for noise removal that it still kind of works right now. So I think we're okay. Just try to speak up.
When I give my lectures, I always forget to put the mic on. But you don't have a mic.
That is the issue. We don't have a mic. There's one sitting in my office that you could attach to your laptop, but this should work sufficiently if you kind of sort of talk to the ceiling.
Okay.
The noise cancelling option should be on the tablet, tablet right there. Is that what you said, Jonathan? It's on the tablet?
Yeah, on that console tablet, there's like a microphone icon, I think in the top right. If you click on that, there's a few options. There's default sound, and then they have a noise removal option, which apparently helps for people online to hear a little bit more clearly.
Filters out background noise. How's that? Any better?
It's still good. You're still a little bit low, so if the best you can, if you could speak up, appreciate it. But it works. It works.
Well, I'll project. How's that?
Yeah.
Okay, I've got 54 slides. Maybe we better get going.
Yeah, we got people still popping in, but go ahead and get started. Dr. Mike Higgins, go ahead and tell us about yourself a little bit and dive in, because I know you have a lot of important and very foundational ground to cover. I'm really glad to have a really excellent teacher here with us. Go ahead, Dr. Higgins.
Okay, so I'm Mike Higgins. I'm an adjunct professor. I teach a course in winter quarter on medical decision making. And basically, the talk I'm going to give you today is abstracted from one of my lectures, actually two of them. A little bit about who I am. I sorry for that long-winded bio I sent out. It's a little longer than I probably should have. But there are two key things I want to come out of that bio, because this can really set the direction of what I'm going to talk about. My origins started, my very first work in medical informatics was actually what we call predictive modeling now. Hal Sox, who ran internal medicine at that time, at the VA, was looking to see if we could find clinical findings that would predict whether or not a patient coronary artery disease. Now, this is back in the mid-70s. So when we got data, we got it ourselves. We didn't download it from the electronic medical records. And working very hard, we would collect data for 500 patients. That was a very large data set, and we built a predictive model. This is a model that we developed, and it's based classic scoring model. Logistic regression is how we got our weights. That's the performance on the training set, and there it is on the test set. It's a pretty good rule. If you like area under the curve, it's an 8.5, that's pretty high. What we were really more interested in is a rule that could rule out the disease. So we were very concerned about false negatives. So the ability to get close to the upper right-hand corner is very important to us. This is a rule we developed. The other thing that was going on at that time, it was very important. Eddie was starting to do research in trying to analyze what should the appropriate cancer screening protocols be. And that's what I'm going to talk about today. He did a study in which he basically showed this was how you would vary an outcome measure, that is the likelihood that the patient would have invasive cancer, how it would vary as a function of how frequently did the intervals. And this is sort of revolutionary. He answered a question that could really not be answered by other means. I mean, ideally, the way we answer questions like this is you do a randomized clinical trial. You get 1,000 patients or 10,000 patients, whatever your budget allows, and you screen one group with one protocol, the other protocol, and we look at outcomes. But if you think about cancer screening and outcomes, you do your studies over decades in order to release the results. And if you think about the technology changes within decades. In fact, we're going to talk about pap smears today. That has changed substantially during that period of time. So you really can't answer these questions by data, but what David showed is that by skillful analysis, you could actually figure that out. That's what I'm going to focus on. The notion that the way we find information, maybe we learn about this complex world we live in, is through mathematical models, and that's the focus of my discussion. His study was fairly controversial when it came out, but so for example, he showed that if you did it every 10 years, you'd get a 64% reduction. If you did it every year, you'd get a 96% If you did every 3 years, you did almost as well. And this was a sort of revolutionary finding because it suggests maybe we should change the intervals for this. More about that later. And to verify as a result, a few years later, the International Agency in Cancer Research, a big European group, did a large study involving millions of patients in which they showed data, which basically verified what David showed. In fact, showed in many respects he was overstating the value of treatment testing. Okay, so here's what I'm going to do. I think I spent a little time talking about cervical cancer. I don't know your backgrounds, but I figured you need to understand the sort of natural history of this disease we're talking about, because that's will be very important in discussion. Then what I'll do is I'll actually use that to show how we can determine patient outcomes, give a change in outcomes, how that leads to a policy decision. And then it's a sort of epilogue. What I'm going to do is I'm going to go to large language models, open evidence, the one will be using, and see how it addresses the same questions. So what's the state of large language models in dealing with the same questions? Okay. So cervical cancer, we're talking about the natural history, and we're talking about how you go about diagnosis. Now, the important thing to understand about David when he did his research, that was back in the late 70s, early 80s. That was before we really understood the central role that the coronavirus played in this disease. It was recognized as something that was involved with us. We didn't really establish until much later. There was actually a cause, probably about 90% of all cervical cancer. I mean, there are other mutagenic processes which cause cervical cancer, but they're probably only about 10%. Most of it's caused by a virus. And here's a schematic which kind of shows what that process looks like. So this is a picture which is showing the epithelium, the layer of tissue that's covers the surface of the cervix. And it's and the sort of normal cells and kind of the sort of neat, orderly. They have layers. The stuff at the top does one function, the stuff in the middle and the top bottom. This is a function. This is a part of our physiology, which serves a function, and that sits on top of the firm tissue and so forth. Now that's what that sort of represents the normal state of this part of the cervix. What happens is the human papillomavirus invades typically at the basal level, and what it does is it embeds itself into the nucleus of the cell. And what it does is then that propagates into the daughter cells of that cell to the normal propagation. Now the virus particle is just residing in the nucleus. It's starting to affect it. It changes its morphology. You can actually see those changes under a microscope. That's becomes very important. We're going to talk about, but it's not really changing the reproduction of the cell until later. Eventually, the viruses actually they take over the genetic process. They actually invade the DNA, and now you have a very different set of cells. These cells lose their function, they change, they look differently, and of course they have the hallmark of cancers, that is they proliferate and grow beyond bounds. That's what cancer is. So we see in this diagram the sort of continuum, normal cells, dysplasia, those deformed cells, confounded to cancer. And the importance of this diagram is the time scale. Going from left to right takes decades. In other words, what starts out as an infection, the time for it to become a cervical cancer is a couple of decades typically. That gives us a chance to find this. Now, the test that is commonly used is a pap smear. It's a pretty simple test, at least as it's initially introduced. You basically use a spatula that scrapes some exfoliated tissue off the cervix. You put it on a slide. You look at it under a microscope. A trained pathologist can recognize dysplasia. That means we can see the harbinger of this cancer before it's actually an invasive disease. And you can see why that's important. And here's a study which shows how well it works. This was sort of started to become a commonly used practice back in the 60s. The 5 Scandinavian countries rolled out their own cancer screening programs to different degrees, ranging from in Iceland, where they screened 80% of the women, all the way down to Norway, where they only screened 4%. And then following up those patients over the next 20 years, they saw this correlation and the reduction in deaths from cervical cancer. Quite a remarkable effect, almost exactly parallels the effect. So this test works. It saves lives But the question becomes, how frequently should you do it? They arbitrarily picked a year. That's kind of what they said, but why not do it every month? Why not do it every day? If you did that, you would find this cancer before it had any chance to be in a lethal disease. Okay. Now, of course, that's easy for me to say because apparently that's not a particularly pleasant experience. But if you just simply look at survival, there's no limit to how frequently you'd like to do it. Why do we do it once a year? It's the sort of question we're asking here. And see, if you look at survival, so the very typical staging of the disease, if you look at the survival rates associated with that, when it's confined entirely to the cervix, either as dysplasia or a local cancer, the survival rates is basically that a patient who doesn't have the disease. These data were collected on 50-year-old women on average. If you look at the 5-year survival rate for 50-year-old women, it's almost exactly the same number. Okay, so that's why it's very important to catch the disease early. Now, David, when he did his work, this was the key distribution that really drove the timing of things. This is the age-specific incidence of cervical cancer. But if you think about it, what we'd really like to know is not the age distribution of cervical cancer, but the distribution of dysplasia. We want to find dysplasia. That's what we want to look for. We want to find it before it has a chance to become cancer. By the way, thing to notice about this is we're talking given the current use of active stimulus, only less than 1% of the women in the world in our country actually will develop cervical cancer. Now, since David did his work, some important studies were done. The Canadians in the 70s and 80s had a policy of not actively following up on positive pap smears. So you could actually see the natural history. They would screen women frequently. If they saw mild dysplasia, they tended to not do anything because most dysplasia self-limits. Our bodies are exquisitely designed to go after these malformed cells. And if you look at the natural history, mild dysplasia, 99% of it will be gone the next time you look at it. But you get up to severe dysplasia, you're still seeing an unstable structure. It drops down to about 90%, it'll still be there. And you can actually see that in the data they collected. This is the problem with progression of cervical cancer for all dysplasia. If you look at severe dysplasia, that's what it looks like. Moderate dysplasia, mild dysplasia, we can see, and you look at the time scale, this is measured out in decades. David didn't have access to that information when he did his analysis. We do. Okay, so how do you determine patient outcomes for this thing? We're going to do is we're going to model cervical cancer progression, and then we're going to see how that affects life expectancy as an outcome measure. We're going to use to model this dynamic disease, we use the Markov models. Now, most of you probably have seen Markov models at some level. Let me just do a little sort of review here. Markov models are based on two concepts. One is the notion of a state. For example, patient initially is free of a human papillomavirus. That's a state. Later on, they become exposed to human papillomavirus. They have an infection. That becomes a dysplasia. That becomes cancer. These are all states. The second key concept in a Markov model is the notion of transitions. So a woman makes the transition from human papillomavirus free to exposure. There's a certain probabilistic description of that phenomenon as it takes place over time. Okay, and then we similarly have transition probabilities between the other states. Now that's the sort of natural history of it. And the other thing to keep in mind, of course, is that women can also die. 25 women tend not to, older women tend to. So we have to have progressions. We have to recognize this and model this. The transition that can happen is if a patient dies, okay? And then finally, there are the probabilities the patient stays in place. Now, this is a Markov model that we're going to use in order to understand what's the distribution by the time period of the appearance of dysplasia. This is what we're really interested in. Now, what we'll see, of course, is the progression of the patient through the entire process, but we're really only interested in the subset of that journey, that is the time it takes to get a dysplasia, okay? So here we go. Where do we get the transition probabilities? Well, these are pretty easy to get. We just get those from the actuarial tables. That's just the probability of dying from other causes, okay? Those numbers are well published. They're a function of the patient's age. If you're familiar with Markov models, what I'm doing now, then doing with a non-stationary model, the transition probabilities change with time. So as the woman ages, the risk of her dying from those various causes increases. That's the nature of our lives. The other transition probabilities describe the progression of the disease are a little more complicated to get. For example, the exposure to human papillomavirus occurs when you become sexually active. So that's the first question. That's the transition from absent to exposure. Then there's the probability that exposure will lead to infections. There have been studies which explore that particular phenomena and so forth. The one number that's hard to get is that number. That is, what's the time between an infection and dysplasia? And you can kind of see why we don't have a lot of observations for that, because we don't screen for infections. And so the idea that we look at infections and then follow them up to see where they're not, there's very little observation. That's going to be a hard number to get. However, if you think about this picture, we have all of the other pieces. Most importantly, we have the distribution for the time to cervical cancer. That's that graph I showed you, the age, the specific incidence of the disease. We have all the other probabilities. So basically what we'll do, the way I find out what that transition probability is a very inelegant method of just cut and fit, see if it fits. I guess what the model is, I run the model, I'm doing monocardal simulation, doing 100 million iterations, so I get a nice smooth curve. And I do that until the cervical cancer distribution matches what we see in the test. And when you do that, that's how I determine. So this is what the curve looks like for cancer, and this is what the curve looks like for precancerous dysplasia. Okay, kind of cool. Now, that's the natural history of the disease that we're looking for. We now want to start asking the question, okay, what does this mean in terms of screening? Here's a function we'd like to know. How long does it take for dysplasia to become invasive cancer? I mean, just if you think about that, I mean, it starts out when you say, okay, dysplasia here, what's the time? Well, there's some probability that increases eventually. This is indeed a precancerous dysplasia. That's a hypothetical curve. How are we going to draw that curve? Well, remember that IARC study I mentioned? If you think about what that study tells us, it tells us exactly the time, it tells us about those time delays. You put in, guess for what that curve looks like, you run it through, you should be able to recreate that curve. And when you do that, what you find is that this curve actually looks like that. It's kind of a squishy, messy looking curve, but it just makes some certain sense. You notice that what happens early on, there's a certain burst of cancer in the first couple of years, but that persists out over time. Now, frankly, to me, there's an interesting question that it raises. Are what we see in these women who are developing cervical cancer 30 years later, is that the same dysplasia that was the indexing event? Or did that initial indexing event go away and we're seeing a new event coming? Those are interesting questions, but empirically they don't. We simply ask the question, when a woman first starts to experience dysplasia, what's the time until they ultimately have cancer? And that's what this curve empirically describes. That's what matches the data. Okay, now the next thing we need to understand is, what are the survival models? We're going to take this from the perspective of a 25-year-old woman. And if you just think about, well, if they don't have disease, that's just the actuarial survival model. If you're 25 years old today, the chances you'll make this 65 is about 90%. Okay, that's what it is in this country, okay? Now, let's imagine what the survival model looks like if at the age of 36, this woman developed an invasive cervical cancer, something they need to treat. That's what the survival curve looks like. That's if they receive treatment. But where did the survival model come from there? That's a whole nother lecture. I'm not going to go into that. But basically, the way I model survival is I say survival is determined by two things. You either cure the malady, in which case the patient returns to the normal actuarial survival curve, or you don't, you die from the disease, in which case your survival empirically typically is exponential. That's a simple model, but it tends to work pretty well. And that's the sort of model here, and I've fitted this using the data. Let's not worry about that. So that's what it looked like if the women had treatment. If they're not treated, of course, they see that exponential survival curve. It looks like that. Now, what really happens, of course, is that eventually it becomes apparent that the patient has cervical cancer. So what you really do is just delay treatment. They're imagined. So you imagine that the cancer appeared at age 36. They recognize it at age 40. They start treatment, and that's what that curve would look like. Now, the reason I pick those particular times, 36 years and 40 years, is that would represent what would happen to that 24-year-old woman if she had invasive cervical cancer at age 36 between her third and fourth exam, because she's being examined every 5 years, 5-year interval, okay? That's what it would look like. And that's what that's what the survival curve would look like if the treatment doesn't start until the patient has their next exam. And that's what the curve would look like if they had their treatment started exactly when the cancer first arrived. So of course, that's not going to happen. I mean, why screen? If the women knew when they had invasive cancer, why bother with the test? But the fact is, there are symptoms that are produced by invasive cancers, and they're not very specific. So there's a lot of things cause those symptoms, but some patients actually do come in and say, hey, doctor, something's not right. So somewhere between those two extremes is what's actually going to happen to the woman if she has an invasive cancer at age 36. What we're going to do for the time being is we're just going to focus on the sort of pessimistic model. That is, since the patient only learns about her disease because she went to her next screening exam and found. We'll come back to this. That is a projection. Okay, so there we have it. Now what we have, that's the survival for a 25-year-old woman if she has cervical cancer that occurs at age 36, okay? There are, of course, a whole bunch of times it can occur during that interval between the third and fourth exam. So we draw that. We can determine the probability of each of those from that distribution curve we forgot for dysplasia. So what we can do is we can take the weighted average of those, weighted by those probabilities, and that's the survival curve for a woman who's 25 if she has invasive cancer found between the third and fourth exam. That's what the survival curve looks like. Okay, let's stretch out the time axis. So you see more of a life, recognize there are many 5 exams. Let's look at all those. We can draw that same curve for all of those other time intervals. Okay, those are the survival curves for each of those time periods. We know the probability of it occurring to each of those time periods, so we can again, from that cervical cancer incident rate, average those together, and there we have. Okay, we have a survival curve. Now that's the survival curve for a woman who will eventually have cancer, and it's not found before it has become invasive, okay? What would it look like if it was found before it became invasive? It would look like the normal survival curve, right? Because this cancer is curable if you find it before it becomes invasive. And the question then, okay, what is the balance of those two? Well, remember that IARC, that study which showed how the probability of detecting cervical cancer is a function of the interval. We look at 5 years. It says there's about a 0.86 probability of doing that. We use that to average those two guys. And we get that curve. And what is that curve? That's the survival curve for a 25-year-old woman who will eventually have cancer if you screen her every 5 years. Okay, if you have a survival curve, you can get all sorts of summary statistics. Let's just use life expectancy. We can compute the life expectancy from that curve and subtract it, and we see what the change is. So if you do 5-year intervals, the life expectancy loss of disease is a little less than 3 years. We can repeat this for different screening intervals, and we get a whole table of effects, okay? Now remember that back a little while ago, so everybody see what I've done here, just survival models and with weighted by probabilities. Now let's go back to that assumption we made a little bit earlier. Remember we talked about using the sort of pessimistic model where the women doesn't go to see our doctor until the next screening exam. What if we said, no, that's not right, that's the worst case. What if we said, no, let's look at the more optimistic case, that's an upper bound. Well, we repeat the analysis I just did using this different survival curve, okay? And we get a set of lower bounds. Do it exactly the same thing. Or if we plot them, that's the upper bound, that's the lower bound. There are the life expectancies as a function of the interval between screening, okay? Now, this is what I determined. This is essentially the same curve that Dave grew 50 years ago. A little different out towards the bigger ones because they didn't have all the little details that we have now at the time. But there's a function which shows us what the effective change in the screening interval is on a patient measure, such as patient life expectancy. Okay, everybody with me so far? Does all make sense? So kind of straightforward, step at a time. So, I mean, when I'm summarizing, it's what takes 2 90-minute lectures to cover in my course. I'm waving my hands a lot here, but you can see how it's 3 minutes all this together. Okay, so here we go. Let's look at policy decisions. Okay. And we're going to talk about cervical cancer screening costs, and then we're going to talk about cervical cancer, the value of life. Now, before we move into the notion of policy, it's important to understand you come up with an answer, like we've just, that graph I just showed you. It doesn't matter until people agree with it, okay? My wife and I knew David when he was doing this research. We would ride bicycles together and send him. We talked with him a lot. He told us a lot of stories. He told us about the hate mail he got when he published this result. One of them came on the form of a official letterhead from a famous institution, and the message written across it in bold red letters was bullshit. That was the only message he got, okay? And that was kind of typical. Here's a trade rag, how they treated this thing. They said, we are very upset by the recommendation. We think it's a great disservice to women, so on and so on. They didn't criticize his assumptions of independence, his distributions. They criticized his result. Okay. Now it's easy to be cynical in the medical profession. They say, because after all, he was basically cutting their customer bases by a third. That's not fair, of course, if you think about it. If you think about the community of people who understand the math that we've just walked you through, and the community of people who understand the clinical implications of cervical cancer, there's not a lot of overlap. So it's easy to see how somebody would say, this is not what I was taught, it must be wrong. That's what was going on. It wasn't until a couple of years later that we began to recognize this is actually a valid way of looking at it. And now if you're familiar with screening protocols, they screen every 3 years, like we recommended. So how do you get to that policy recommendation? So here's the remaining lifetime for a 24-year-old cervical cancer patient. In other words, if you think about screening, the only beneficiaries are the patients who eventually would develop the disease. All of the other people you screen, they're just part of the cost. They're not part of the benefit, okay? And these are the benefits that you see. These are what you see for different screening intervals for the woman who will have cervical cancer eventually. If you want to get the total cost of the screening program, it's a pretty straightforward present value calculation. It looks kind of messy here, but the piece is all pretty straightforward. First of all, the patient has to remain alive, so you have a survival function in there. That's pretty straightforward. You have a cost of the pap smear test. I just said $50. That's kind of what it is some places. It's more out in a place like this. One of the costs of screening, of course, is you do have false positives. This test is pretty good. It has about a 90% specificity, but there's still about 10% of the cases come back positive. And when they look again, there's nothing there. But when they looking for it can be very costly. So we're going to assume that about 5% of the positives, of the false positives require a test. And we're going to say that test is basically a biopsy, a $1,000 procedure, okay? So that'll give us a cost of procedure. There's a discount rate, 3%. And then finally, if you think about it, in order to really understand the total cost, you have to divide that by the incidence of cervical cancer, okay? And when you do that, what you find this table, depending on what your interval is, okay? That's what it costs to screen to find that one beneficiary. Okay, that one woman who enjoys that increased life expectancy. To plot that cost versus expectancies, you get a curve that looks like that. Now the policy decision is where do you operate on that curve? Where do you pick how far do you go? This is where I think the world gets really messy. I'm not very comfortable with what we do next. But see, what you do basically is you do incremental cost effectiveness ratios. If I increase the interval, decrease the interval from 10 years to 9 years, I have an effect on the benefit, I have an effect on the cost. And that's what these ratios are. We have two rows here because we have an upper and lower bound effect because of that confusion and uncertainty about how the thing is treated. And if you look at what the three interval, it basically says you're willing to accept about a $200,000 for a year of life. That's about the cost, that's about the break point. And is that a reasonable number to use? Now, economists will give you this really detailed ways they go about determining statistical value of life. When I look at it, it all looks like nonsense to me. They're making some terrible assumptions. But you can still take this number and compare it to other things we do. This is from a paper that looked at 500 different cost benefit analysis. And these are the sort of numbers that you see, the cost effective ratios, dollars per year of life. And you can see that's kind of sitting down there. It's kind of a high end, so they're kind of consistent with that. And that's how they make policy decisions, okay? Now look, what I want to do now is I want to move on to large language models, okay? In particular, first of all, I downloaded ChatGPT 4. I do that because I give out exams, and the exams are all take home. I want to make sure I'm asking questions they can find the answers for on the internet, okay? Recently, our offspring, who's going to school at one of those other schools in this country, learning to be a doctor, turned me on to Open Evidence. And Open Evidence is an exquisite large language model. It's very good. It's really fun to work with. So I'm going to show you some answers, how we answer some of these questions. So first of all, I say, let's think about the cervical cancer screening interval. How frequently should a 24-year-old woman be screened with pap smears for cervical cancer? And this is what it says. It says for 25-year-old women, risk blood every 3 years. Points out they actually should include human papillomavirus screening, something I completely left out. This discussion, what you'd really do in a real analysis, and then gives, it's a very good answer, but what is it doing here? It's not solving that problem. It's finding the answer by doing a search. That's not what we're looking for, okay? However, it's awfully good at finding stuff. Like, for example, I had a lot of questions. I spent a lot of time finding that data. And what's the average time between cervical cancer dysplasia following the onset of human papillomavirus infection? It gives you a pretty good answer. It gives you essentially the same answer that I found the hard way. Okay, that's pretty good. So that's a pretty good answer. Then I ask it, what's the time from the onset of precancerous dysplasia? Time for the onset of precancerous dysplasia. There it gives me an answer. It's got nothing to do with the question I asked. It's kind of frustrating. It's a hard thing to do like this. So as a source of populating the numbers, pretty useful, but you kind of have to be careful of it. How about it in dealing with sort of analysis, figuring things out? So first of all, I'll say, well, first of all, what I really like to know is what is the time interval? What was the life expectancy for a woman screened every 3 years? And it gives me this very nicely written, entirely correct answer, but it's entirely a qualitative answer. I want a numerical answer. You know, so how does it do analysis? How does it do an analysis? Let's start out with sort of survival models. Let's use the actuarial survival model. That's the survival model for the normal population. What is the life expectancy for 25-year-old women in the US? And it comes back and it gives you exactly what the correct answer should be. And that's not too surprising. It just simply finds the correct actuarial table. Good at that. What if I gave it a model? What if I gave it a constant risk survival model, the exponential? What's the life expectancy there? It recognizes that correctly, and it gives me the correct answer, 10 years. Cool. And then what if I put those two models together? So I want you to do a little calculation. I say, look, suppose a 25-year-old woman is going to go into treatment. If successful, she'll live a normal life expectancy. If not successful, she'll face a 10% mortality rate every year. What's her life expectancy? And it does a very fine job of answering that question, okay? Gives you exactly the correct answer. So that's kind of impressive. Now, I was asking this as a follow-up question. I would ask one question, then I do a follow-up question, and so forth. I then stepped outside of that stream of questions and asked it just as a new question. And it came back and said, I can't answer your question. And what was happening there, it was kind of subtle. I didn't say the woman lived in the US. And it treated that as ambiguity that it didn't give an answer. I think that's cool. If you can't, doesn't have to, shouldn't answer the question. If you would put in there, if you put in there the woman lived in the US, it gives you the same answer as before. Now, and so forth. Now, let's start with some of the other calculations, present value calculations. That should be pretty simple. So I say, okay, I've got a screening thing, that sort of present value calculation I showed you a few minutes ago. Can I do that? And it comes up with the correct formula, gives you an answer, but the number is wrong. And if you look very closely, you can see what the number is wrong. It can't compute powers. It's off by the powers. It's interesting, when you go to ChatGPT, it's even worse. For some reason, these guys can't compute the power of 0.103 raised to a power. It underestimates the value. Can't understand why that's true, but it's true. Okay. Now let's give it a Markov model. Okay. Let's give it a real simple Markov model. We say the patient initially is in a state in which they have a 1% chance of dying each year, 10% chance of dying each year. There's also a 50% chance there's stable change where they face a 20% chance of dying each year. What's their life expectancy? That's the simplest Markov model I could think of with any sort of substitute. Now, I've left off a lot of the response. It does recognize this as a Markov model question, even though that didn't have a word in the equation. That was kind of cool. But it didn't start saying all sorts of interesting things. It gives us this recursive relations. It solves the recursive relationships. It comes up with this answer like this, and it says the answer is that that's complete nonsense. It's got nothing to do with how you solve Markov model questions, if you're familiar with this. The actual answer is quite different than that. I don't know where that came from. It's simply made up. And this is open evidence, a very good, it is completely screws up on that. And then I give it my classic, what I call the canonical decision problem. This is where I'm going to have utility in the analysis. So I say, like, suppose a treatment has a 1%, a 10% probability of causing immediate death, but will give you an annual mortality rate of 0.2 if you survive, or you have a 20% chance of dying and you only have a 10% annual mortality rate. And furthermore, I say I have risk aversion, I have a utility function, so I don't treat all lengths of life the same. They use a commonly used model called the exponential. Ask it that question, and it was cool. It really recognized all the details. It even came up with the right formula. That's the integral you solve that problem. Everything is really cool. I said, great. However, it then says that, does that simplification solve that integral? That's the wrong answer. You see, actually that integral is 1 minus the number they said. Now, when I saw this, maybe I'm paranoid, but I'm not a doctor, so I can't normally subscribe to open evidence. So I wrote them a message, because I teach this course and I'd like to use your problem. Your permanent literature fine, and they gave me a license. About an hour later, I get an email from Open Evidence. Turns out a former math student at Stanford was very interested in the course I taught. So we corresponded, kind of changed. And then I asked this question, and I'm left wondering, was he actually watching me? And when he saw this question come up, this is a very human type of mistake. See, he has, instead of having lambda over lambda plus m, he has m over lambda plus m. I mean, I've made that mistake. That's a human mistake. I can't imagine an algorithm. I mean, if anybody who's used Wolfram knows that the computers can solve integrals just fine. It couldn't solve the integral, okay? And I'm kind of suspicious. Also, they have a lot of references. Maybe one of the references I'm referring to is wrong. But the fact is, it got the wrong answer to this in a very peculiar way. Okay, so here's my conclusions of this long-winded talk. Mathematical analysis can provide answers to important questions. I mean, can you think of that? I think that's very clearly true. Data search, research, the stuff that an awful lot of our colleagues here at Stanford are involved with, will provide useful value codes analysis. We'll learn more about the uncertainties and the outcomes of diseases. And furthermore, large language models are awfully good for finding those. I mean, it took me a week to find numbers that it found in 30 seconds. I wish I'd had that tool when I was building the models, okay? And it has but currently large language models have very limited, and it cannot do analysis. Now, I emphasize current. I think a fool's is anybody who believes it's not going to get better, but it's not there yet. It cannot do the analysis. Okay, that's it. So that's what any questions, any comments?
I don't see any questions online right now, but any questions from the room?
Was any of this a surprise to anyone? See, part of my motivation for giving this lecture is I don't know of anybody else at Stanford teaching this material. They teach it over in management sciences, Rush Ackner and those folks, very good at probabilistic analysis, but teaching it in the medical domain, I'm the only one knowing this. I'm aging out. I'm not going to do this forever. Somebody needs to be teaching this.
There's something striking about this whole topic, and that is there's so much of what people do in medicine, assuming that there's evidence behind what they do. And I think what was striking about David Eddy's work is that suddenly it poked holes in what people assume were reasonable practices based on some kind of information. And now we realize so much of what we do is just based on culture.
Yeah, but yes, yeah, that's true. However, I think the field has moved forward. So in the late 70s, early 80s, David did a whole series of cancer studies. Cervical cancer was just the first one, the one he used. He did breast cancer, colorectal cancer. He also, he was here at Stanford, at Duke University, lured him away by giving him an endowed chair, plus 5 other endowed chairs to populate with a department he was going to lead. So he went there and hung out for a while, but then he discovered it was much more fun just doing consulting in this area. And a lot of people are interested in screening questions other than doctors. For example, people running railroads want to know how we should inspect the trucks. That's a screening question. And so he had a whole career. It spread out. And in the meantime, we started taking cancer screening questions seriously. And PCOR, I would argue, that's a patient-centered outcomes research institute. These are people who are systematically looking at the evidence-based medicine. But yeah, if you think about the gap between understanding the medicine and understanding this mathematics, it's still quite a displacement, it's a separation. And one of my missions is trying to bring those two together. I get a couple of graduate medical students every year in my class. I always cheer that.
There's a question by Mary Goldstein. Mary, you want to ask her a question?
Oh, okay, great. Well, first of all, Mike, thank you for this really interesting talk. So the first question is just a quick informational one, since you've looked so much into this and I don't happen to know. There are women who develop cervical cancer in their early 20s and obviously haven't had decades of exposure after HIV. So are those cases always within the 10% of cervical cancer that's non-HPV related?
That's an excellent question. And I was wondering that myself. The only study I've been able to find on that 90% did not break it down by age. They just simply, they sequence the DNA of tumors and they find human papillomavirus in 90% of them. But I don't know about the distribution. It's an interesting question.
Okay,
Yeah.
and I had a couple of other kind of quick, I'll try to be quick, so I'm sure lots of other people have questions too. So and I'm not practicing currently, and it's been a long time since I did women's health, so I'm not speaking from, any current clinical expertise. I'm speaking more as somebody who just looks things up to sort of see what's going on in the field. So the standard recommendations for pap smears usually say something like for women of average risk. And I wonder if you've done any analyses for women who are at higher risk or women who are at substantially lower than average risk.
No, my read on that qualifier they put in there is they're basically saying if you've had a positive pap smear, you're in a different risk group. I think that's what they're saying. And also it's the way how do you factor in human papillomavirus screening, which is another part of the screening test they do. So I think that gives you different distributions of risk. But no, I haven't seen anything beyond that.
Right. Okay, and then this will be my last question. So I think in many practices, HPV screening is now done routinely. I don't know what the recommendation is, but I know it's often done. So I wonder if that provides any additional information. And related to that, I think the current recommendations, if you look them up in some standard sources, they're really complicated. And they're based, they include a reference to when was the last pap smear and was there a normal pap smear within the last 5 years. And they also include something about the HPV status. And then people also make refinements depending upon which HPV variants, because there are some that are a higher likelihood of risk for cervical cancer than others. So I'm wondering if, beyond this 1 hour talk, I'm sure you're doing an awful lot of work on this. Are you exploring any of those other things and, looking at these very detailed current recommendations?
And to be honest, no. See, to me, this is an interesting application of mathematics. I'm not a policy wonk. I'm not trying to set the policy. I'm trying to understand how you can solve a complicated question. What you said though is very important. There are about 200 different varieties of HPV, and only about 50 of them are thought to be related to the development of cancer. And the policy, if I remember it correctly, says basically every 3 years, if that's your only test. If you couple it with human papillomavirus screening, you do pap smears every 5 years, and that's that complication you're talking about. If you're really worried about when you should be screened, go read the recommendation. Don't listen to me.
Well, there's also the what to do when something is found on the pap smear, and those which would when to do a colposcopy, when to go right to a biopsy, and those get very detailed with respect
Yes.
to the recency of the last normal pap and what the HPV was, and so on. So a lot. So whether the person, how quickly they will get treated and whether they will be effectively treated, it comes into play there.
Yeah, and that's interesting because you see one of the diagnostic maneuvers is actually a very good therapeutic maneuver. The cone biopsy removes the tissue you're looking to see how far it's gone, and if it's not very invasive, you've also removed the tumor. Now that particular procedure has a complication rate, there's a higher risk of miscarriage afterwards and stuff like that. It's not a procedure to be treated lightly, but it is a curative treatment.
Right, but even to whether to send someone to colposcopy, like some cases of abnormal pap, they don't even do a colposcopy to have a look, let alone do a biopsy. So anyway, it's a very complex area on what happens after an abnormal pap smear.
Yes.
And I'm sure there's mathematical analysis that could be done on that too, but that's way over my head. Anyway, thank you so much for a great talk.
What's great about cervical cancer screening is that it's actually the only preventative screening test we have. It's exactly as you're saying.
Yeah.
So most screening tests screen for the presence of cancer, whereas in this case you're screening for the precancerous lesion, and that's one of its amazing attributes. The cost thing's very interesting because I've worked with HPV for a long time in the past, and I'm not up to date as to what's happening now. The cost issue is interesting because it only matters if you're actually going to do something if you detect an abnormal pap smear, right? So you wouldn't screen unless you were actually going to go and do something about an abnormal finding.
Yeah, but as you're saying, this is, if you think about the Canadian study, they had a large population of women who had severe dysplasia that they said, well, let's just wait. And now they don't do that anymore. It was like 60% they would proceed, they would do further testing on. Now it's like 90% even in Canada. But that's sort of letting it sit there. And the fact is, because our body's awfully good at eliminating malformed tissue. To
actually have the natural history is fantastic.
Yeah.
And I was trying to think another way for you to figure out from the HPV infection to the detection of dysplasia in terms of actually being able to work on that parameter. The
it would be interesting to see now that we have HPV vaccine, Canadian girls all get HPV vaccine. You could actually do a comparison, historical comparison of the amount of dysplasia you're not finding compared to the amount that was found over that same number of years. So that might give you
No, that all makes sense. And that's an interesting question, because you see, in a way, we've already seen that effect. Because if you think about that incidence, only 0.0065 is the prevalence of cervical cancer in American women. That's not a major cause of death. Way back before it's pap smear, there was a much higher incidence of it, but we don't see as much cervical cancer because we find more dysplasia.
Right.
You remove that. Yeah, and that's an example of how it changes in some of these. And then, of course, the vaccine, of course, I can imagine that 100 years from now, nobody be interested in this question. You know, it'll be like chickenpox. Imagine that.
Bumps.
Okay. That's it, right? Any more questions?
Just my question is about the market model you mentioned at first in the beginning of the slides. I would like to, in that market chain model, you calculate the probability of each transition and also the deaths. I would like to see if does it make sense to also consider the probability of getting treatment or, for example, getting vaccine, and how this market model will change based on this criteria.
And that's straightforward. I mean, Selena could tell you all about that, but that's straightforward. I just think my model was focused on a single question: what's the time to dysplasia? That's all I was concerned about. For example, you notice I didn't show people that with cervical cancer dying. There was no branch from there from dying, even of course they do die. That's the issue of the disease, because I was just simply trying to find the distribution for the pathways from 25 years to dysplasia, and you could have put in there a chance of treatment, yes, and that would just become a branch, and you had another leg going off from that. Yeah, markup models are extremely powerful tools. You can represent anything. Yeah, I'm a big believer in them.
So you mean it makes sense to add, for example, and it may actually will make accurate our model by considering this exposure as well.
You see, in the power markup models, it goes even beyond that. See, so you if you have a dynamic, uncertain system, stochastic system, we can draw this diagram of it, and we can draw these very complicated diagrams. Back in the days when we solved Markov models analytically, which you can do if you're clever enough and it's simple enough, we were sort of limited. But now when we use Monte Carlo simulation, you make that model as complicated as you want to capture all the little nuances. You can say, well, I'm not sure what the transition probability is. It's a range. Each iteration of your Monte Carlo simulation, you pick a possible value of that range. You have to run it 10 times as long, but you can still even deal with the fact you can be uncertain about the parameters and still get a distribution, which is, it's a very powerful tool. We believe in that. Thank you.
There's two more questions online. Do we want to do one more?
Sure.
Geoff, you want to ask your question?
Hi, Mike. Jeff Rutledge. Great talk. Thanks very much for going into the details. We could go back and forth about the clinical details related to cervical cancer, but I'm curious, do you think, is it your understanding that this methodology, this approach to analyzing the appropriate screening intervals is being applied? I mean, my impression from the recommendations of the USPSTF, the Preventive Task Force, is that there is this kind of thinking happening. And had you looked at the other obvious applications, which include colorectal cancer screening, where, you know, early colonoscopy can define early precancerous lesions, which determine the frequency of follow-up testing, frequency for value testing.
Jeff, you're exactly right. This is, I'm not the only practitioner of this method by a long shot. No. First of all, David Eddy started a whole company. So this is a very actively used tool. There's no question about that. My only concern is I want like Stanford to retain a center for learning. But no, it's a very widely used tool. Yes.
for sure. Thanks, Mike. Great analysis and lovely level of detail.
There's also a question by Dalibor about use of ODEs or nonlinear models in this field. Is there a perspective of using that in this field?
I don't quite understand the question.
So the question is there potential to look or model this from the perspective of dynamical systems, ODEs, nonlinear dynamics, and simulations?
Yeah, I would argue that's what I've done. I mean, what those words can mean to me is a Markov model with time varying transition probabilities. That's and I guess some and Sunita maybe have a sense of this. There are the Markov models with the transition probabilities are all constant forever. There are Markov models in which you allow the parameters to change. Then there are semi-Markov models. And I don't see and I think maybe there are questions about semi-Markov models because I don't see the difference in a semi-Markov model and a transition and a standard Markov model in which you allow the transition probabilities to change over time. Gives you the same sort of behavior. That is the uncertainty about when a transition occurs. You no longer think of it as discrete, but as a random variable itself.
And Jonathan is also saying that Doc Owens is teaching this with Jeremy Goldhaber. That's what he's comment. Jonathan, you want to say something?
Sure, I'm trying to walk the streets of New York here, but that Doug and Jeremy have definitely taught on this subject. I don't know if they still are. So there are people continuing the tradition, and I think the USPSCF has used these kinds of models. I wonder if I can squeeze in a broader perspective for you, Mike, when you think about this. There's so many companies out there, they got early detection for cancer, they're gonna check your blood, all this kind of stuff. And can you point out how I think it's almost always fundamentally fraught when you're trying to detect rare disease in asymptomatic patients? And do you have to insist on 100% sensitivity or not? How do you think about it, given that you've thought about this rigorously?
Oh, shoot, I don't quite understand the question. Can you ask it again?
Isn't it fundamentally difficult to impossible to have a screening program for rare diseases in asymptomatic people? I mean, you can try, but a lot of companies try to do that. And I think a lot of them are going to be in trouble when they deal with the numbers.
Yeah, but what do you call a rare disease? Is less than 1% a rare disease? That's cervical cancer. And if you do the numbers, if you move that zero over a couple places, you're now paying millions of dollars to save that 1 year of life. Yeah, it stops making sense. Yeah.
But I guess what you're offering is a explicit approach to calculate when it is quote unquote worth it. And another common complaint I sort of hear people is like, well, with such a bad disease, anything you do has to have 100% sensitivity or nothing. How do you address that? You're really talking about incremental cost effectiveness ratios. And that gets very fraught where you pick that decision threshold. How should people think about that?
Yeah, but you see, if you think about, if you're looking at the benefit, you're only interested in the false negative rate. Okay, if you're interested in the total cost of the program, you're also interested in the false positive rate. The false negative rate was actually incorporated into the analysis, if you think about it, in order to projecting what the benefits are of the treatment. Then when you say, okay, now we have benefit of this one woman with this imperfect as the current stated sensitivity for pap smears is about 97%. That's a complicated story. Water-based preparation. It's way up there. But yeah, so you already reflect that in the benefits to the women. But if you say, okay, now I'm going to roll this program out for enough people to benefit that person, that's where you see the cost. Now you're interested in the false positive rates because that adds to the cost of the test. Also, I don't know of a test with perfect sensitivity. Do you have one?
I don't, but I've often argued with people who insist that anything we do has to have that, otherwise it's unusable. And I try to tell them that is irrational, but human instinct, emotion doesn't feel good about it.
Yeah. Yeah, there's a lot of craziness in this world.
Great.
Thank you very much.
Thanks, Mike. This was a great discussion review, and anybody who can should try to take Mike's class when they can, because there's a lot of foundational reasoning we can all learn. Thanks, everybody. See you next week.
Okay, thanks, Jonathan.
I'm seeing. There we go.
Take it big.
I think John was talking about the class.
Yeah.